It's all time series. I mean, almost all time. We have some, let me see here, we have some spatial data as well. So what this is probably the most complex experiment. This is a time series, right? So every one of these is a neuron, and the colors represent different neuron types. So unlike an artificial neural network, the brain contains lots of different cell types. And this method, you record them inside the brain, and then outside of the brain, later you identify lots of different gene RNAs, and that lets you tell which cell is of which type. So this is spatial data. And there is other types of data than time series in neuroscience. So there's this sort of thing which is transcriptomics, where you've got the locations of the different genes around the different cells in space. And there's connectomics, which is how the different neurons connect to each other. So it's basically a graph, which neurons connect to which other neurons, and you get a graph from it.
I thought you needed to kind of kill cells to measure.
Yeah. So you get the time series in vivo with this 2-photon microscope. So this is it in vivo in the living brain. But then after the end of the experiment, it's a mouse. You remove the brain, and then you do this experiment. But that's ex vivo.
So when you do an experiment, is the mouse moving or is the mouse not moving?
There's both types. Yeah, it can be either. Moving, just for technical reasons, means you have smaller number of cells you can record, because this device, this 2-photon microscope, is the one that can record 100,000 or 10,000. You can get one that's small enough for the mouse to comfortably carry on its head, but it's nothing like as powerful. So you get maybe, a few hundred neurons simultaneously that way, but you can get like 50,000 this way. But then the mouse is still awake and it's still able to do things, but the head is held in place. So they do behavioral tasks. So here's an example. They like basic video games. They sit in front of a screen and turn the wheel. It's literally a Lego wheel. Turn it left or right, like a kind of a driving game, to move this grating into the middle. And this is in reality, this is the only part we can actually randomize, because you have the stimulus can appear on the left or right. That's about the only thing we can randomize is what the stimulus is the mouse does. Or if we present sensory stimuli, like another thing you often do is you show the mouse visual images, so it sees thousands of visual images. You could present those in a random order. You could present random images, but most of what you're interested in is things that the mouse does itself. Like it decides whether it's going to turn left or right. And we want to find out what's going on in the brain, what's different before a left turn and before a right turn, because that's kind of the command of the movement. Yeah, we're just chatting.
Is it the task just trying to experiment design to figure out how the brain works?
Yeah.
Respond to different tasks.
Well, okay. So the biology question is how the brain works, and we're able to record tens of thousands or nearly 100,000 neurons simultaneously, and their activity is going to give you a lot of clues, but we need to then make the mouse do different things, right? So see different stimuli, perform different actions. We'd love to get causality from the brain to behavior. I don't even see how that's possible. Although you can manipulate brain activity. I'm not going to talk about that here, but there's a technique called optogenetics where you can stimulate or silence neurons at will. And that you could get causality from the brain to action that way, but that's not what I'm going to talk about here. What I'm going to talk about is the recordings of large numbers of neurons, and the only thing as an experimenter you then manipulate is things like the stimuli that the mouse sees. So the causality would be going from the outside world into the brain, rather than from the brain to the outside world, where you can do that. You can manipulate the brain activity. I should have thought to talk about that today, maybe, but I didn't make any slides on it at least. Yeah, it's sort of the reason I'm here is to ask for help, really. I mean, so I thought I'd make that clear. I'm not like the aim of presenting these methods is I want you to think, God, you're doing that. You could do much better than that. Like that's kind of my ideal outcome.
So I know neuroscience is quite broad.
Yeah.
Are you thinking the kind of neuroscience you do?
Yeah. I mean, well, in general, really, but particularly what I do, which is the recording of the time series.
Yeah, what you do is quite sophisticated in terms of the kind of data generated.
Yeah, but
Well,
it's
I know some people also do like high throughput. I mean, they know biologists here who kind of work on the fundamental biology of.
like the cell biology. Yeah.
Cell biology.
Yeah. They've all got tons.
Experiments tend to be less sophisticated. They also get data.
They get, yeah. The problem with our experiments is that it's not just up to us what happens, it's up to the mouse too. And so we can't really control what we want to control.
Makes sense.
Yeah. So we've,
Does neuroscience cover like psychology?
yeah, some would include that. Yeah, yeah. So and these problems are very much still there in experimental psychology. You know, so.
So your official title is experiment.
No, quantitative neuroscience is my job title. But there is, I think it's Cambridge as a Department of Experimental Psychology, or maybe it's Oxford. One of them have.
Department of Psychology.
Yeah.
Experimental psychologist there.
Yeah, which would be, you know, you get humans to do similar tasks with these sort of, you know, you see this stimulus and you move left or right, or, you know, things like that. They all have the same issues. Those tasks have the same issues as ours, you know, in terms of autocorrelated time series. And again, they probably just don't think about it at all. I'm going to show an example, though, of how you can use activity in the mouse brain to get a statistically significant prediction of the price of Bitcoin, which obviously shows there's a mistake in that method, right? If you use a naive method, you can predict Bitcoin prices from the mouse brain.
Yeah. Some people in our field who also talk about this nonsense correlation.
Yeah, exactly. That's exactly the word. Exactly the word.
There's so many nonsense correlations.
Exactly, nonsense correlation. Exactly the word. Exactly the word I use.
I have the impression that like a lot of statisticians who work on neuroscience, they work on Bayesian method.
Yeah,
These
yeah, yeah.
bias or-
Yes, they're very popular. I'm not a massive fan, to be honest. I mean, it's okay. I just don't really understand them as I don't know. I don't trust them as much. Basically, the problem is, this is my problem with Bayesian methods, is if you have a Bayes factor between two models, one of them's terrible and the other one's really terrible. Then the one that's terrible wins by a massive Bayes factor.
I see.
right? And that's all you get is everything's relative between models that are, they could all be bad.
Just tells you you're doing something wrong. It won't give you any information about your target. But it tells you that something's wrong. Would it be useful?
Can you do that?
Perhaps.
I don't know how to do that. Okay, that's
Your base predictably optimal, but if your model is specified or your classical model is specified, it won't collapse just before. It will just give you a lot of uncertainty and tells you something's wrong.
but does it, I mean, like if you're comparing two hypotheses, two discrete hypotheses that could both have parameters, you've got priors for your parameters in both hypotheses, and you get a Bayes factor between your hypotheses, what will this method do in that situation? That that one of them's terrible and the other's really terrible. Do you know what I mean? That's my problem with Bayesian methods is if you're distinguishing between two really bad methods, two really bad models, but one of them's even worse, then you get the one that's only really bad is a huge winner, and you get a huge Bayes factor for it.
Shows you that something's wrong. One is both are the only
Yeah. Yeah.
one would be less smaller than the other, but both will be.
Yeah. If there's a way for it to tell you that. But if there's something else that tells you that, the Bayes factor doesn't tell you that.
Yeah.
Something else. You're right.
I don't know.
We should. Yeah.
So we get started.
Yeah.
We can keep it very.
Okay. Right. Yes.
Thanks for coming.
All right. Well, thanks for having me. It's great to be here. As you can see from the title, it's sort of a cry for help. We've got all of this amazing data, really incredible data. We don't have the methods we need to draw valid conclusions from it. Okay, so what I'll talk about is first the data, or some of the types of data we have, then the statistical problems we face, and then some current approaches. And my goal here is for you to say, these methods are terrible, you could do much better than that, and then tell me how, or start working on it. So that's kind of the goal. All right, so first quickly about the data. This is about recording from the brain. The biological question is how the brain works, how we make decisions, how we process sensory information, how we plan what to do, how we combine sensory input with plans to make decisions, all that sort of stuff. The brain contains billions of neurons, and we can now, in mice, record up to around 100,000 of them at once, which is a tremendous opportunity for really figuring out how the brain works. All right, what does the data look like? So I guess I can't assume anybody knows what neurons are. So neurons are cells in the brain, and they talk to each other through channels called axons and dendrites. They're like a neural network, I mean the original neural network, but unlike an artificial neural network, they use a pulse code. So a neuron in an ANN has a real valued output. Neurons in the brain have a pulse code output, so it's discrete action potentials, which are a point process, right? So you've got the activity of the brain is an extraordinarily multivariate point process. You've got 1 billion cells, and they're all firing these discrete events at certain times. That's what the data looks like, and that's what we need to analyze, and we need to correlate it with the sensory stimuli that the subject is seeing or hearing, and the actions that the subject is making. And if we can draw causal conclusions, but even if we can't, we want valid correlational statistics would be great. All right, so there's two main ways to record these brain activity, and I'll go through it quickly because I really want to move on to what you do after the recording. But the two ways are, the first you can record with electrodes, or second you can record with calcium imaging. Electrodes have a very high temporal resolution, or at least compared, the clock speed of the brain is about a kilohertz. It's ridiculously slow compared to a computer. So 1 millisecond time resolution is about all you ever need for determining the times of these action potentials, or spikes as we call them. But with electrodes, you get that time resolution, but you only get around 1,000 neurons tops. Calcium imaging has a lower temporal resolution, around 100 milliseconds, but you can't see the individual action potentials. You just get an average of all of them, a filtered version. All right. So electrode recordings, first done here, I think in Cambridge in the 1920s by Lord Adrian, was the first to record the action potential with an electrode. But nowadays, we have something more advanced. These are called the neuropixels electrode. They're made in a semiconductor fabrication plant in Leuven, Belgium. It's got an integrated circuit on top, and then at the bottom is the actual recording shank, which is very long. This is an electron micrograph. It's got all of these recording sites, one after the other, and this goes into the brain, originally of mice, but now also done in humans too. Incredible work, recording in the speech cortex, and it's possible to predict what somebody's going to say from the activity in their speech cortex. In any case, originally in mice, it's got all of these recording sites, and they go next to the neurons, and when the neurons fire an action potential, you pick up an electrical signature of it here. This is what the data looks like, the raw data. It was connected by Nick Steinmetz there in the lab Matteo and I have at UCL. This is what the raw data looks like. These are time series. You can see the time axis here. That's 10 milliseconds. So all in all, we've got about 100 milliseconds here. And these are the raw signal from these different channels. And you can see a lot of noise, but also these discrete events here. These are the signatures of the spikes. We want to go, first of all, in pre-processing from this raw data to a point process. That's not what I'm going to talk about today. It's a challenging problem, and we still need better solutions for it, but it's not what I want to talk about today. Today, let's assume that we've detected the times of the spikes. So these colored lines are the times of the detected spikes. And you can see it hasn't got all of them either, because the method isn't perfect, but it's got some of them. And now we want to understand the times of these. This is where the point process comes from. So if we zoom out, that was just a few of the channels you record from this neuropixels electrode. We're zooming out in space over more channels, and we're zooming out in time. And now you can see what this multivariate point process starts to look like. The different colors are different neurons that have been recorded, and the tick marks are the times that they fired. And we're now looking at a few tens of seconds. I think eventually we're going to get down to a minute. But you can see what the complexity of the data is starting to look like. This is still nothing like the full data we have, though. So here's, again, this is about 10 years old, this data. So there's much bigger data now. This is two of these neuropixels electrodes in the brain of a mouse. And it's recording, close to 1,000 neurons. Now people are getting maybe 5,000-10,000 with a lot of these electrodes at once. And it's in different parts of the brain. And now we're zoomed out so much that you can barely see the individual spikes, the individual action potentials, but you can start to see some things. So at the top there, that dashed black line is the time of a visual stimulus that was presented to the mouse. And here are the recordings in the visual cortex. And they're actually, when you look at it this way, you don't see much sign of the visual stimulus. It's there, but it's subtle. What instead you see is these sort of events here. You see them with your naked eye. This is a part of the brain called the thalamus. It's in the middle. It's considered the brain switchboard. And these almost certainly corresponds to time the mouse is moving, right? The mouse is making a movement, and you have this increase in the firing rate, the intensity of the point process in the thalamus, and you have a decrease in other places like here, the motor cortex. This is one of the things that you know, it's just what the brain does. There's a lot about movement in there. The correlations, biggest correlations you get in the brain is with the movement of the subject, because that's what the brain is for. It's for controlling the muscles. If you didn't have muscles, you wouldn't need a brain. All right, so that's the first way of recording with electrodes. The second way of recording is optical, and it uses 2-photon calcium imaging. So the neurons are cells, and if anyone's done the biology, you'll know that the way the action potential happens involves sodium coming in and potassium going out of the cell. But as well as that, when the neuron fires an action potential, calcium comes into the cell. It normally has a very, very low concentration of calcium because it pumps calcium out. But when there's an action potential, calcium comes in for various reasons, biological functions, but that's not our concern. Our concern is it gives us a signal that we can optically measure when the neuron fires an action potential. So this here is a 3D molecular structure of a protein called GCAMP, which is a calcium-dependent fluorescent protein. It was engineered ab initio using a fluorescent protein from a jellyfish as a template. Some people engineered it so it's now calcium sensitive. And it's a protein, so it can be put in the genome sequence. And there are organisms, mice, worms and flies and others, that express this protein natively in their neurons. So, and you look at this mouse, it looks like a completely ordinary mouse from the outside, but actually in the brain of this mouse is this calcium-sensitive fluorescent protein that when the neuron is active, it becomes fluorescent in the green color. All right, the other part of this technology is the 2-photon microscope, which is a way of getting activity, of measuring the activity deeper into the brain. So with an ordinary microscope, the focal plane looks like this. It's never really very focused, but a 2-photon microscope uses illumination light of half the normal frequency, so infrared, and it's so strong, it has these incredibly strong femtosecond pulses that it causes fluorescence of 2 photons being absorbed at once, which gives you an incredibly sharp focus of this tiny dot compared to that big beam there. So what that lets you do is record in 3 dimensions the activity of these cells, and they're flashing away. You know, if you watch an old movie from the 1960s about how the brain works, they would show the neurons flashing to symbolize their activity. But now they literally are flashing because they contain this fluorescent protein. When they're active, they flash, and we can get about 10,000 of these or more simultaneously and extract their activity. So what you see here is now it's a pseudo color plot where we've got the x-axis is time. It's a much slower time scale. This is 100 seconds now rather than 100 milliseconds. And the y-axis is neuron number. And the intensity of the gray shows you how active that neuron is. So at these moments here, you see we've got some cells that become more active and others that become less active. That's the time that the mouse starts running, which you see in that trace at the top. It's running along here, then it stops, and then it starts again, then it stops, and it starts again. And you've got these stripes of activity, which some neurons get more active, others get less when the running happens. That's one of the main, again, the main correlate in the brain is to do with activity. But we want to know exactly what neurons relate to exactly what activity. Another thing we can do with this technique is use post hoc molecular methods. This is a way of detecting different RNAs which identify different cell types that let us put this in color, because now we know what the different cell types that make up these circuits are. So unlike an artificial neural network where you only have one type of neuron, in the biological brain, there are hundreds of types of neurons in the cortex, and they all do different things. So you can see that the ones that were active during running are one type, and these ones are suppressed during running. They're another type of neuron. And understanding that those differences is going to be a clue to how the brain works. Okay, the last piece of experimental data is about behavior, right? So if you want to know, what we really want to know is how the brain turns a sensory stimulus into an action. If you're going to do that, you need to have a situation where the mouse is turning a sensory stimulus into an action. And this is a task that was used by an organization, the International Brain Lab, which was a worldwide collaboration of 20 labs or so who recorded from the entire brain of the mouse, covered the whole thing systematically with these neuropixels electrodes. And it was all using the same task, and the task is illustrated here. You've got this mouse, and it's sitting in front of a wheel. It's literally a Lego wheel. It's like a little video game. This grating appears on the left or the right, and the mouse has to turn the wheel to drive the grating into the middle, and then it gets a reward. But if it turns it the wrong way, it gets a burst of noise like that they don't like. So this is the timing of it, which turns out to be quite important. So you have to have a pause, and then there's a sound at the same time that the video stimulus comes on. They need the sound because it helps them, you know, perk up and notice that there's something on the screen. And then they're allowed to move the wheel. And if they move it in the right way, they get this reward. If they move it in the wrong way, they get this loud sound. One of the key things for the statistics is what we can randomize and what we can't randomize. So one thing we can randomize, and pretty much the only thing we can randomize, is where on the screen this stimulus appears. Does it appear on the right or on the left? Which stimulus do we present on any trial? That's something we control. For this particular task, the International Brain Lab task, there was more than that because they wanted to go beyond just how the mouse responds, but also to have something about how the brain incorporates prior knowledge. So in this case, the y-axis of this plot is the probability that the stimulus is going to appear on the right. So it starts 50-50, and then it goes down to 80-20. That means that 8 out of 10 trials, the stimulus will be on the right, and 2 out of 10 will be on the left. And then it flips to 20-80. So now 8 out of 10 times it will be on the left. And then it flips back, and then it flips back again. And it flips, the block flips also at random time. So we've got 2 levels of randomization. First, we've got the blocks, which flip at random times. And then within the blocks, we've got the stimuli with a probability, which is conditional on the block side. And here you see that the mouse's behavior tracks these blocks. And if we look at these are called psychometric curves. It shows the fraction of right choices they make as a function of the contrast, meaning how bright the stimulus is on the right or on the left, and it's colored by the block. So what you see, if you look at the 0 contrast ones, that's the time there's no stimulus at all, just the sound, and the mouse has to guess which side it is on. The right thing to do is in the block where it's normally on the right, to choose right, and the block where it's normally on the left, to choose left. And we see that at 0 contrast, we've got this separation, right? So when the stimulus really is, when we're in a block that the stimulus is normally on the right, when there's no visible stimulus, the mouse will normally choose right. And the other way around, when the stimulus is normally on the left, the mouse will choose left. So it's not just responding to the sensory stimulus that it sees, it's taking into account some prior knowledge about where the stimulus is likely to be. And that's the sort of thing we want to understand how it works in the brain. We now have probably the data to figure that out, but we don't yet really have the right statistical methods to do it with a real certainty. And that's why I'm here. So these are the sort of questions we want to ask. Okay, so one of the other things about these behaviors is you can't always randomize everything, right? Ideally, you would randomize everything that's not nailed down, but we can't do that. We can't randomize what the mouse does, because that's not up to us. It's up to the mouse. And if you want to correlate brain activity with the mouse's choice, well, that's literally going to be observational, because we can't randomize either of them. We can't randomize the choice, and we can't randomize the brain activity. There actually are techniques to control brain activity called optogenetics, which is when you shine a light and it makes the neurons active or suppressed. So there is some way we can randomize brain activity, but we're not going to talk about that here. We can randomize the stimuli that we present to the mouse, but even that is often incomplete. So for example, often the stimulus you show on one trial depends on the mouse's behavior on previous trials. So for example, if what these mice often do is whatever they get in a mood where whether the stimulus is on the right or the left, they just pick right every single time, regardless of what the stimulus is. I don't care, I'm going right, I'm going right, going right. And eventually, with the idea that, well, eventually it's going to be on the right and then I'm going to win. So I don't really care, I'm just going to keep going, yeah. And then I get my reward anyway. So what you do is you make it that when that happens, the stimulus is always on the left, right? So if you're going to keep choosing right, well, then I'm going to keep choosing left until we come to some sort of understanding. Okay, but what this means is now that the stimulus has to depend on the prior choices. So it's not completely random. It's conditionally random. It's conditionally random, conditional on the past, but it's not totally random, and that makes the randomization tests hard. Okay, you can randomize, but conditional on the past. All right, so let me tell you now, switching over to the statistics, what are the problems that we face? And the problem, I know I just heard this word used in this same room, the problem is nonsense correlation. So I guess you may know this paper by Mr. G. Udney Ewell, CBE MAFRS, delivered to the Royal Statistical Society in 1925, almost 100 years ago. And he makes the point that if you plot this graph, which shows both two things on the same time x-axis, the first is mortality rates, and the second is the fraction of marriages in the Church of England, they both are very strongly correlated. They both go down steadily with time. They're very well correlated, R equals plus 0.95. But this is a nonsense correlation. It's not just that there isn't a causal relationship between these two. There isn't even really a correlation. But the question is, how do you define what a correlation means before you can test it? So we need to first have a rigorous definition of what we actually mean by a real correlation, not a nonsense correlation. Then we need to try and find ways to test it. All right, so nonsense correlations is our problem in neuroscience because everything we record depends on time. The mouse might suddenly start to decide to keep going right, and normally they do that at the end of the experiment when they're bored and they just want to get out of here, like, are we done yet, sort of thing. And so the behavior changes towards the experiment, end of the experiment. Also, the brain activity changes, but that doesn't mean they're related. It's just because it's at the end of the experiment. You've got two variables that both depend smoothly on time, and they're correlated with each other, but only because of a common time dependence. How do we correct for the common time dependence? What's often done is things like taking a residual prediction from the past. You make a prediction from the past, you take a residual. There are two problems with that. The first is that it makes all sorts of assumptions of linearity which aren't going to be true. But the second, that's even worse for us, is that we're not even talking about numbers. We're talking about point processes. You can't subtract a residual of a point process because you can't add and subtract a point process. The decisions to the left or right, you can't add and subtract them. So what do we do how do we correct for time dependence and nonsense correlations if we're not even looking at numbers, never mind it being non-linear? It would be great if we can make claims about causality, and maybe sometimes we can, but even the much more limited goal of having valid methods to detect correlation, that would be a big step forward if we have really rigorous methods for that. All right, so let me illustrate. Here's a trace where we're predicting the motion energy from the mouse's brain. So here's a recording of brain activity. Time is on the x-axis. I'm sorry, there's no axis label. I think this is probably about 1 second all in. No more, maybe tens of seconds. And they're arranged so that the neurons that correlate best with movement are at the top in red, and the ones that correlate negatively are at the bottom in blue. And this is the actual and predicted movement of the mouse. So there's a video camera that's measuring how much it's moving, not just the wheel, but like twitching whiskers, wiggling the nose, anything like that. And you can't see any difference because they're right on top of each other. You can really predict incredibly accurately the actual motion of the mouse from a linear sum of this activity, just a weighted sum, ordinary linear regression. And if you do an F-test, you get p equals 2 times 10 to the minus 84, incredibly significant. The other thing people often do is cross-validated prediction, right? So this is 10-fold cross-validation. Each one of these blocks, we use the other 9 as the training set, learn the prediction and evaluate it in this block. And we still get an R of basically R squared of 0.8, very good prediction. All right, so that's all good. All right, but here's the problem. This is the exact same analysis used to predict the price of Bitcoin from the brain activity of the mouse, right? So you do exactly the same thing, and you get actually an even smaller p-value predicting the price of Bitcoin from the mouse brain, which is obviously absurd, and it's because they're both temporally autocorrelated, and that's all it is. So this is the problem we face, and the cross-validation doesn't solve it either, right? It doesn't look quite as good anymore, but we've still got an R squared of 0.7, a very high cross-validated value when we predict cross-validated the price of Bitcoin from the mouse brain. And obviously the mouse doesn't know the price of Bitcoin. So, all right, so how are we going to deal with this? And here's what we're currently doing. And again, I'm not making any claim that these are the best things to do. They're just all we really know at the moment. Okay, so first, what does it mean, the null hypothesis, that they're not actually correlated? So what we have to do is think that we combine all of the data in the experiment, all of the recordings of neurons, into a matrix X, right? Which is a number of time points by number of neurons matrix. It's a random matrix, or you could flatten it into a random vector. And now let's suppose that we're predicting not the motion energy, but the binary block is the stimulus in the left. Is the stimulus 80% chance to the left or to the right? You remember we had these slowly varying binary blocks. So this would be a binary random vector of length number of trials, right? So we've got 2 random vectors, one random matrix of size neurons by trials. The other one is a random binary vector of size number of trials. So the data for a given recording session is just these 2 things together. And our null hypothesis is that this matrix is independent of that vector. The whole matrix is independent of the whole vector. So there could be correlations between neurons, and there could be correlations between times, and there could be correlations between behaviors at different times. That's all fine under the null hypothesis, but there can't be correlations between any one of the neurons at any time and any one of the behavioral time point choices at any time. That's our null hypothesis. And what we really want to do so this is a property of the probability distribution over these pairs. And what we really want to do is draw an inference about this probability distribution. Like, are these two things independent of each other? We want to draw that inference from a single observation, if we can. So if we have the hypothesis that this matrix is independent of that vector, can we tell that just from one observation of each? And we can under some additional assumptions. All right, so what's the first way we would do it? Well, the first way is just a plain old randomization test, but we give it a slightly fancier name. We call it the pseudo-session test. And the reason is, if you've done an experiment in which some of the experimental variables were randomized, you can use a randomization test. It doesn't require independence across trials. It doesn't matter if the stimulus is autocorrelated. So in this case of the block, right, we had this this block that at random went, this random binary vector, and we can redraw it from the same distribution. Redraw other samples from the same distribution. These are both autocorrelated, right? This is autocorrelated. That's the problem with chalk. And this is autocorrelated. But it doesn't matter because we're redrawing the whole sequence of these behavioral block variables. And we can ask, does that correlate with the neurons? Does the actual data correlate with the neurons better than the null ensemble of resampled? So let's have a look at that. So here we've got the neural data. So this is a raster plot. Red means that this cell fired a lot of action potentials. White means it didn't. And this is the behavioral block variable. And we make a test statistic, which is just the difference, which is that we're trying to predict this binary variable from this vector. And if we predict the actual block variable that the mouse actually saw, we get this accuracy. But if we redraw random stimuli, we get this null distribution, and we're way outside of the null distribution, which means we do have a significant coding of this block in the brain. To validate it, what we can do is we can take, I'm sorry, that wasn't the block, that was a stimulus, whether the stimulus is on the left or right. To validate it, what we can do is we can take brain activity just before the stimulus that shouldn't depend on the stimulus, and then we do the same thing, and now we find we don't have a significant effect, right? We shouldn't have had that significant effect. But what we need to do is use the actual distribution that generated the stimuli rather than just an independent variability. All right, so that's the first thing. And the next one, which is also pretty simple, is what we call the session permutation test. So the pseudo-session method needs randomized experiments, but we don't always have that. So if we're correlating the brain activity with the behavior, we don't have anything randomized. If we're correlating the neural activity with the choices, we don't have anything randomized. Neither of them was randomized. Both of them are autocorrelated. But what we can do is we can take account of having multiple sessions of the experiment and do a permutation test. Ask if the behavior in one session is more similar to neural activity on that session compared to the others. So we've got our brain recording on session 1 and our behavior in session 1, same on session 2, 3, etc., all the way to session S. And we compute a test statistic which measures the predictability of this from that, and we sum it over the sessions. And then we generate a null ensemble by shuffling, right? So we shuffle or permute the sessions that we're using to predict the the behavior, right? So rather than predicting the behavior on session 1 from the neurons on session 1, we predict them from the neurons on session S. And this gives us a null ensemble, which is S factorial values. It's too many to compute all of them, so we take a random sample, a random set of permutations. Our data is the full histories of the neural activity and the behavior, or the stimulus and the behavior. We get a test statistic for each session, and we add them to get a global test statistic. Then we generate some random permutations of the sessions, and we get a null distribution where we're comparing the actual neural data to the permuted session. And then we use the usual formula to get a p-value, the number of times the actual statistic exceeded the null distribution. Okay, and let's look at this. These are the recording sessions from the International Brain Lab again. And we're comparing a prediction. I think we had 5 sessions here, so there's 120 possibilities, 5 factorial. And we're comparing the prediction of video motion in the actual session from the brain activity, and it's better than all of the permutations. But if we do the same thing now to predict Bitcoin prices, now it's right in the middle, right? As you would think, the Bitcoin that was actually simultaneous with the neural recording had no more in common with it than Bitcoin at any other time. It was just a nonsense correlation, and we've got rid of it with this method. Okay, those are all pretty standard. This one, as far as I know, is new. And this is what you would do if you've only got one session and you can't resample, you need to make further assumptions. And so we make the assumption of stationarity. So the definition of stationarity is just that the probability of time-shifted data is the same as the probability of the original data. And it's a property of the ensemble, not of any one instance. It's a probability of the distribution that generates these time series. But if we assume stationarity, we can draw conclusions about the ensemble from any one instance. So here's how it works, is you've got your 2 time series, and you take a central portion of one of them, and you have a test statistic that measures the correlation of this with the other series. And then you take the same segment and you shift it from the beginning to the end, and that gives you a null distribution. And this was, it's in this preprint and also independently from Yuan and Xu a few years later. It turns out you can't actually just use this shifted stuff as a null distribution, because there are pathological counterexamples, which really are pretty crazy, but they do exist, where the shifted distribution, if you just compute the percentile of the actual correlation within the shifted distribution, it'll appear to be significant even if the null is true. But what you can do, so the 2n+1 of these shifts, right, where n is the size of the stuff we left out, you would think the naive test is if you're in the top alpha times 2n+1 of those. Actually, if you make sure you're in the top alpha n+1, so you basically have double the p-value you would do otherwise, it's still valid, it's conservatively valid. If instead you take the normal 2n+1 p-value, it's valid in the limit that the length of the time series goes to infinity, but it's asymptotically valid, but it's not exactly valid for a finite length of series, whereas this conservative one is valid even for a finite length. All right, so this is showing it now on the brain activity. Here we've got the actual, right, here we've got the prediction of video motion of the mouse from brain activity as a function of the shift. And you can see it's all around here, and then it has a big peak at 0, because predicting the actual simultaneous brain activity is much more accurate than predicting the brain activity shifted a little bit in either direction. And if we compute the p-value, we find that we get something very significant. The actual value is far outside either the conservative or the approximate distribution. We get strongly significant in either case. If we do it for Bitcoin, then, as you might think, the brain activity is no more correlated with the simultaneous price of Bitcoin than it is with the price of Bitcoin a second before or afterwards. And we get just a flat, noisy curve, and we don't get any significance with this method. All right, there's something a bit more complicated. So now we come to the situation where the stimuli are randomized, but only conditionally randomized, where, like in the case where we have to keep putting the stimulus on the left if the mouse keeps choosing right, that sort of thing. How are we going to deal with the case that if we want to do a randomization test when the experiment was only partially randomized or only conditionally randomized? So sometimes when the stimuli depend on actions, you can derive a conditional distribution. So an example of this is this particular experiment. I won't go into the details. Imagine it was a human and a light comes on and you need to press a button, right? But you could just cheat by pressing the button all the time, right? So to stop that happening, if you press the button before the light comes on, the light's not going to come on. And it's only after a certain time of you not pressing the button that the light comes on. And now we want to know, can you actually see the light rather than just pressing the button at random? And it turns out that in that case, you can derive the conditional probability of the light time conditional on the actions, and you can use a conditional randomization test that is exact you've got one statistic that's observed of the reaction times, and then you can find an exact distribution for the conditional distribution of the stimulus time given the behavior, and then you can do a randomization test with that conditional distribution. That sometimes works, but it doesn't always work, and when it does work, it's normally quite hard to find those conditional distributions. So something that's more general that you can use is the idea of history conditional independence. And again, as far as I know, this is a new idea, although it's really nothing that complicated, and it's in this preprint. So often, the probability of a stimulus or a reward or something that you control, it depends on the history of previous choices. So you can't randomize the entire sequence. And if we want to test the hypothesis that the entire sequence of stimuli is independent from the entire sequence of brain activity or choices, we can't do that. In this case, for example, that the stimulus repeats on the wrong choice. But, so you can't use any of these methods like pseudo-session or session permutation or linear shift, because they're testing for the independence of the whole history of stimuli from the whole history of neural activity. And we know that's false before we even start, because it's a conditional independence, not a full independence. But what we can do is we can define history conditional independence, which is saying that given conditional on the history up until the moment t, conditional on that, the brain activity and the stimulus are conditionally independent, right? So now that's the null hypothesis we want to test. And there's something that you can do, which is called a tangent sequence randomization. Tangent sequences are something that comes up in, the theory of time series statistics, it's just usually used as a proof technique, but you can actually use it to make a statistical test as well. A tangent sequence is when you've got a time series and you know the conditional distribution at every time given the past. You redraw a random value at time t, conditional on everything up to t minus 1. And that's your value of the tangent sequence at time t. And then you forget about that, and then you replace it with the actual observed value at time t. And now you draw a distribution, you redraw a sample at time t plus 1, conditional on the observed values up to time t. And you can keep doing this, and you'll get a random sequence. It might be a sequence that couldn't actually have occurred, right? Because you're drawing these samples from the actual observed distribution, not your randomized distribution, right? If you sampled each time point conditional on your randomized distribution, you'd get something drawn from the original distribution, but you're not doing that. You're sampling each time point conditional on the observed up to that time point, and then you're forgetting your random sample and you're drawing it again, and you keep drawing it again. That's a tangent sequence. It's normally just used as a proof technique, but you can use it to make a randomization of your stimulus sequence, and that does give you an approximately valid test. The reason that tangent randomization does is because it approximates something else, which I'm going to call the martingale z-test. So a martingale or a martingale difference sequence, as I imagine you know, is a random process for which the conditional mean at time t plus 1, conditional on the history, is 0, right? It doesn't mean they're independent of each other. It doesn't mean they're uncorrelated with each other. It just means that the expectation at time t plus 1, conditional on the past, is 0. Right, so if you can get from your data, which has got this partial randomization, this conditional randomization, if you can define a martingale difference sequence, and you can also calculate the history conditional independence, which you normally can, right? Because this is all about the stimulus you decided to present on time t, and you know the conditional distribution of it. And this test statistic x might be, for example, the correlation of the neural activity at time t and the choice at time t, the stimulus at time t minus the stimulus conditional mean. In that case, this is going to be true by definition if the neural activity is independent of the stimulus. And you're going to be able to calculate what this is. All right, so if you can define a martingale difference sequence like this, then you can apply the martingale central limit theorem, because the sum of a martingale difference sequence is a martingale. And the martingale central limit theorem says that as time goes to infinity, and as you rescale by the cumulative conditional variance, the value of the martingale converges to a standard Gaussian. There's a wrinkle, which is that you can't stop it at a particular time. If you stop it at a particular time, you can imagine pathological counterexamples, like the best of which is the original double or quits game, which led to the definition of the word martingale, where this test becomes invalid. But if instead you fix a variance threshold and you say, I'm going to stop when the variance, the cumulative conditional variance exceeds a predetermined threshold, and this is large enough, then you define this z statistic, and it should be approximately Gaussian, so you can use a z-test to test whether this is true. And so that gives you an approximate test in the case where you only have conditional randomization, not full randomization. And here it is for the International Brain Lab data. So CT is the mouse's choice at time t, left or right, and ST is the stimulus at time t, which is left or right, and S bar of t is the history conditional expectation of that stimulus, and that's not 0 because the stimuli go in these blocks. And this is a martingale if the choice on every trial is independent of the stimulus conditional on history. And so we can simulate it here. And what we see in this case, this is the cumulative conditional variance of the martingale. And this is the actual value of the martingale in a simulation where the mouse can't see the stimulus. And this is the same things in a simulation where it can see the stimulus, right? So what we've got, we fixed a cumulative variance threshold ahead of time, and we stop it there, and we see, this is the cumulative variance threshold. We fix that ahead of time, and we always stop it here. And if this value is greater than this is the 95th percentile of the Z statistic, then we get statistical significance. So we get it here when the mouse can see the stimulus, but not where it can't. Okay, so that's okay. And it turns out that doing this tangent sequence randomization, where you redraw at every time from this history conditional distribution, is equivalent to the martingale Z-test because these conditional x's give you this they approximate the sum conditional variance. All right, so there's not really any need to do tangent randomization after all that, because you might as well do the Z-test. All right, finally, the problem with that is, I mean, that's good, but it's an approximate test. It's not, and it would be great if we had an exact test or a test that was true even for finite samples. And this is where we use the paper of Zhang and Zhao, who had this really remarkably obvious in retrospect idea, but only in retrospect, that if you have a sequence of p-values which are sequentially conditionally valid, so each one is uniformly distributed, only conditional on the previous history, then you can just use Fisher's method to combine them. So even though they're only sequentially conditionally valid, you can still use Fisher's method. That's my understanding of what you said, basically. All right, so can we use that? Well, actually, no, we can't, because our p-values are generally, if we're doing a one-trial p-value, a conservative one-trial p-value, when we've got a stimulus that might appear on the left or right, our p-values are normally things like 0.5 or 1. And getting a whole lot of 0.5 p-values is significant. But Fisher's method won't call that significant. If you combine a whole lot of 0.5s, even 100 0.5s in a row, you won't get significance with Fisher's method. So what's the solution? The solution might be post-randomization. Post-randomization is when you draw now, given your p-value, you draw something that has a marginal uniform distribution on 0, 1, because it's uniform between 0 and 0.5 and between 0.5 and 1 there. So it's marginally uniform. And then you will get lots of small values. You'll get a lot more statistical power from this. So you can still combine them with Fisher's method and get a valid statistic. But the problem is that we don't want a post-randomized p-value. We want a deterministic p-value. We want a p-value that's a function of the data. And the reason for that is that as an experimental scientist, if you you submit a paper to a biology journal saying, we did this statistical test that generates a random p-value, and I promise we ran it only once, and if you run it only once, and not if you run it 100 times and pick the best one, but if you run it only once, it's a valid p-value. And the reviewer is going to write back and say, yeah, sure you did, right? You ran it 1,000 times and you got significance once. You expect me to believe that? Rejected, right? So we can't do this. Post-randomized p-values, they're great for statisticians, but for a working scientist, they're just no good. We can't use it. We need a deterministic p-value that's a deterministic function of the data. So Here's a solution to this. Given any post-randomized p-value, you take its bth quantile, right? So for example, its median would be its 0.5th quantile, and you divide it by b. So in other words, twice the median, or 10 times the 1/10th, the 10th percentile, or 100 times the first percentile of the post-randomized distribution. That is a valid p-value, and there's a proof here that I'm not going to be able to explain on the fly, but it's in a preprint that hopefully will be uploaded very soon. Okay, and here's some empirical results for it. This is from another part of the International Brain Lab data. This is the moment in training the mice. It's just for one particular moment where we do this thing of if they keep choosing right, we keep putting the stimulus on the left, right? So we have to use some sort of conditionally valid test for that, and simulated this post-randomized Fisher method and some other methods. One is running Fisher's method on conservative p-values, 0.5 or 1, that's never going to give you significance, and you can see it never gives you significance. Mid p-values, it's another thing from the literature, which is to basically take the middle of an interval. And here we've got Rubin-Delancey's bound, which is just from another paper in the literature, and we're comparing two cases. This is the case where there is no stimulus, so the mouse shouldn't be able to see it, so you shouldn't get significance. And you don't get significance above the stated value except for this Rubin-Delancey method. But then here's when the mouse should be able to do it, and this is the number of times we get significance at these different alpha values, and you can see that for all the alpha values, this method performs certainly better than the conservative p-values, which never works, but it also performs better than these other values. All right, so to summarize, traditional methods don't work on correlated time series. Randomization or conditional randomization works if you have a randomized experiment, but conditional randomization is really hard to get working in practice. Session permutation works if you have multiple experiments. Linear shift works if one time series is approximately stationary. Incidentally, people in neuroscience usually use circular shift, but that doesn't work because it introduces a big discontinuity, and then you can, circular shift tests the null hypothesis of circular permutation invariance, which of course that isn't true. The martingale-Z test, or this new determinized Zhao-Zhang method, should be good to test history conditional independence, but we really need your help. Thanks. Oh, great. You came all this way. Wow.
I have a quick question in terms of your data. So you said there is a U notation which has X, right? So
Yeah.
X is for all the records of the, X is N times T, and N is the number of neurons, and T is the number of observations.
Yeah.
And then later you showed like a experiment for like one of the tests that only has like a binary choice for the mouse and the binary choice of like where we put the stimuli.
Yeah.
So for both of these examples, I guess like are they using essentially the same data? That's like you just change the X from this matrix to like.
Yeah, I've flipped about in the applications, in the examples of, in some cases it really was neural activity, and then that's multivariate, right? You've got n neurons at t time points. In other cases, it was just, does the choice correlate with the stimulus? So then X is also univariate, right? But
in that case, do you want to adjust for anything, or you just want to see like the marginal like correlation between the choice of the mouse and the
Yeah. So great question. And the point is, we want a method that doesn't even care if your data is univariate or multivariate, right? So the point is, let's just think of y as being a time series of anythings. They could be scalars, vectors, discrete, continuous, or point processes. And x is another time series of anythings. And the point about this is that the null hypothesis we often want to test is that the entire sequence of x is independent of the entire sequence of y, right? And for all of the tests except the history conditional ones, that's the null that we're testing. And it doesn't matter whether x and y are univariate or multivariate or discrete or continuous or anything. They're just time series of anything at all. And for the history conditional ones, again, it doesn't matter, right? As long as we're able to define a martingale or a history conditional p-value, then it doesn't matter what this data is. All we care about is that it is a time series.
I'm personally working on a
Aha.
Ranger causality testing work with a professor here in Cambridge. So our method can work perfectly for history conditional independence testing you were talking about. But the problem we have right now, not like a problem, or kind of like the restriction is really we can't deal with like a very high dimension x. Like if you're we allow t goes to infinity, but like if you're like the first example where x is n by t, like the n right now we can't really deal with like a very high dimension n, for example, like the number of neurons, but potentially like a subset of neurons. But the y now we're only looking at like a one time series, basically univariate time series. So we're like now looking at the Ranger causality question, like whether the history of x can predict the future of y given all the history we've had right now. So I wonder if like any of your example would be like benefited from this.
Are you saying that you'd like to test your methods on my data? Well, yes.
Would it be possible?
Absolutely. Absolutely. Right. Let's talk back in London.
Why do you test this marginal independence of activity of neuron behavior block? I mean, presumably the behavior are driven by the neuron activity.
Well,
Not that often, so.
yeah, so yes, but now we're going to do this for particular populations of neurons, right? So we're going to ask what parts of the brain and what neurons do you find this significant effect and what you don't. The answer, for what it's worth, is it seems there's some regions that have it more than others, but in every part of the brain you may find one or two neurons that encode this sort of thing.
Okay. Slightly changing my questions. You have these kind of three things. You give the stimuli, which you can control and randomize.
Mm-hmm.
Then you measure the activity of the neuron and behavior of the
Yeah,
neurons.
yeah.
So I can understand how you could use a randomization test to say the stimuli have the causal effect on
Yeah.
certain brain regions, certain groups of
Yeah.
neurons, and perhaps on the behavior as well.
Yeah.
But I thought partly, at least in this slide, you were saying you were also interested in how the neuron activities determine the
Yeah.
behavior.
Okay. So two answers. The first is, if we can even get a valid measure of correlation without even claiming causality, then we'd be quite happy, right? I mean, like asking, does the neural activity correlate with the choice, independent of whether it causes it, even being able to validly ask that isn't so easy because they're both autocorrelated time series. So, we have the correlation between brain activity and Bitcoin price. If we can't get rid of that, then we can't really say anything at all, right? So just to be able to say that we've got a method that says brain activity correlates with movement, but it doesn't correlate with Bitcoin price, even that's something we want to achieve. Looking into the future. Right. Okay, so to that part, there is a method, and I haven't talked about it today, that you can use to control brain activity. This is called optogenetics. It's another type of genetically engineered protein, and it looks a bit like this again. It's a channel. It lives in the cell membrane of the cell. Only whereas this one is fluorescent under the control of calcium, the other one is sensitive to light. It's called channel rhodopsin. Originally, it's from a unicellular organism. And when you shine light on it, it now lets sodium into the cell, which makes the cell active. So you can shine a light on the brain and drive a population of neurons at the time of your choice, and therefore at a random time. You can also, using other methods, you can silence neurons at a time of your choice. It's less powerful because in the standard methods, you don't get to control which neurons you activate. So you can really just silence a whole region of brain at a time of your choosing and at a random time. There are some very difficult new experiment methods where you use, this is already a combination of about 5 incredibly complex techniques. Another thing to make it even more complex is you have this all going through a holographic projector, a spatial light modulator that then can make a 3D hologram inside the brain. And you can use that with your optogenetics to silence randomly chosen neurons and then ask causal questions of which neurons are driving their behavior. And one of the frustrating things is that the people who've spent years getting this incredibly complicated laser hardware together to do this now aren't all doing randomized experiments. And several times I've had a conversation with one of the people who's doing these experiments, and I say, how do you pick which cells to silence? And they say, oh, I just pick them. Like, what they should do is, pick 100 cells and then randomly choose 20 of them to silence, then they can do randomization tests. And I always say this, and they always go, oh yeah, that's a good idea. But, that it's like, people just don't think in terms of doing randomized experiments, even when they can, which is really frustrating. Well, after that, yeah, but like it's really shocking how few people in biology as a whole, I can only really speak for neuroscience, but I'm sure it's true in the rest of biology and psychology, just don't get it about randomized experiments. I mean, for a drug trial, everybody knows you have to randomize it. But when you've got something more complicated like this, where you're choosing neurons to silence out of, or you're choosing trials, which is going to be a right and left stimulus, people just don't think in terms of these randomized experiments. It's really frustrating. I didn't until, about 5 years ago, and when we started getting results that were absolutely absurd, like things that just shouldn't correlate with each other. The Bitcoin was a bit of a joke, but other things like that just shouldn't really be true. And you're like, wait, why am I getting this result? And then you realize that it's because you're using an invalid test. But most of the field just hasn't had this realization yet.
Sounds like you should write an opinion.
I did in 2020. Yeah.
Sorry, thank you so much for the great presentation. The data looks very exciting. The first one is actually a quite tough question. Can we go to the place where you want to use Fisher's combination test to aggregate the results?
Which one? Oh yeah, at the end.
Just a clarification question. So the plot for your individual p-values, are they always flip-flop?
No, not always. No, I'm just saying if we chose a stimuli at random, but 50%, often they're 0.2 and 0.8. But the point is just they're not going to be 0.003, right? Because we're randomizing out of a small number of discrete possibilities. And the chance of, and none of these things are really conditionally rare, right? So if we take conservative p-values, they're never going to be really small, right? They might be 0.2, but they're not going to be ever individually less than 0.05. And I think the limit, like I did a calculation once of what p-value you need when you give loads of them in a row for Fisher's method to give you significance. It's something like 0.3. If you have p-values above 0.3, it doesn't matter how many of them you have, Fisher's method is going to converge to a p-value of 1.
And then, so the reason I brought it up is like, if all the t-values are put on 5 or 1, they're like ways based on binomial distribution to aggregate those p-values even more efficiently.
Yeah, I mean, I think if they were all literally binomial, then you could do that.
Yeah, it's not always 0.1 is essentially just binomial.
If it was literally always 0.5 or 1, then yes. But it's not literally that. I mean, sometimes it's 0.2, sometimes it's 0.8. There may be a way to do it based on the discrete. There probably is.
Yeah, because it's like independent things adding up. So, like, if it's not binomial, it will be not too far away.
Is there a way to, if you've got, basically we've got a sequence of, what do you call it when you draw one of n samples, one of n possibilities, multinomial? Okay, we've got a sequence of multinomial, draws from multinomial distributions, and on every trial, you can have a different set of multinomial probabilities, right? That's the data we're working with, is like on trial n, you've got a set of possible multinomial outcomes, and that set of outcomes might be history dependent. And then we've got the predicted probability of each of them under the null, and then we got which one happened, right? That's what our data looks like. And any test that works for that is good. And the martingale one, I think that we know about. This one, where you make these post-randomized p-values, that I think ought to be valid. But if there's a way to do it that doesn't involve any post-randomization at all, that would be fantastic. I just don't know how to do that for the generic case of multinomials where it's different probabilities on every trial.
Simulate you have a way of combining them that you can simulate this distribution, drawing from this multinomial.
Even though they're all history dependent, right? So the point is on trial n, for one history, you have one history, and then on trial n, it might be 0.5, 0.5, and we got that one. And then for a different history, you might have 0.2, 0.8, and we got that one, right? And so not just this choice, but even the actual probabilities is dependent on the history, right? If there's a way to do that, that's fantastic. I just don't know what it is.
When we were having this Poisson trick, we can have them up and down distribution. It could be pretty easy.
Yeah, we can think about it.
Sorry, I didn't understand. Poisson what? We'll talk about it afterwards. Okay.
Yeah, you'll be here for a.
Yeah, I can stay around. Yeah. That'd be great.
Question is, so there's a literature doing exact inference for bandit algorithms.
For what?
Bandit?
Bandit. Yeah. That's very much like this. Yeah.
That type of technique. There's a professor previously at Harvard, currently maybe at Stanford, called Lucas Jensen. And then Rena also has been on that. Right, so I wonder.
I don't know the paper, but let's discuss the technical things.
Again, that sounds fantastic. As long as it works when the probabilities of the bandit are able to switch in time.